My name is Natalia Tretyakova. I am a distinguished McKnight University professor at the Department of Medicinal Chemistry and the Cancer Center at the University of Minnesota. We are fortunate to have NIH funding for 5 major projects. The main project that has traditionally been the focus of my lab is investigation of the chemical modifications of DNA. So what we do in this project is we use organic synthesis to synthesize non-canonical DNA bases and nucleosides that are formed as a result of chemical exposure. We then use solid phase synthesis to generate small pieces of DNA containing these unnatural nucleobases. We then use the tools of molecular biology to incorporate the synthetic DNA into cellular DNA. Then we can work with cell culture to investigate the outcomes of these chemical modifications in cells. So this work enables us to understand the chemistry of DNA damage and also the mechanisms of how it affects the cellular function, such as DNA replication, transcription, and to establish repair mechanisms of such damaged bases. In the second project, we're focusing on the origins of inter-individual differences in cancer. So, for example, in people who smoke, there is a large difference in lung cancer incidence between different ethnic groups. African American smokers and Native Hawaiians are at much higher risk for development of lung cancer as compared to Latino and European Americans. So we hypothesized that these differences in lung cancer risk originate from genetic and epigenetic differences that are observed between these ethnic racial groups. So we are using the tools of analytical chemistry and epigenetics and also genetics, such as genome-wide association studies, to understand how these different ethnic groups metabolize tobacco carcinogens differently, leading to much higher risk in specific groups of individuals compared to others. This is very important because we want to understand which smokers are at high risk for the development of lung cancer, so that these individuals can be targeted for smoking cessation and chemoprevention therapies.
So when humans are exposed to carcinogens, these carcinogens are metabolically activated to reactive electrophiles, which can then go on to either form DNA adducts or can be detoxified and excreted from the body.
In the Tretyakova group, we are interested in 1,3-butadiene, one of the most abundant carcinogens found in tobacco smoke. Occupational exposure to butadiene also occurs in the polymer industry and has been shown to result in an increased risk of leukemia. In addition, lab animals treated with 1,3-butadiene develop tumors in multiple organs. 1,3-butadiene on its own is not carcinogenic, but is metabolized by CYP450 enzymes to reactive epoxide species such as epoxybutene, which react at the N7 position of guanine to form DNA adducts such as Epiguanine 2. To test our hypothesis that the balance of bioactivation and detoxification of butadiene plays a role in the ethnic differences in lung cancer risk, we have developed high-resolution mass spectrometry methods for the detection and quantification of butadiene DNA adduct Epiguanine 2 in human urine. Among smokers, we see significantly higher adduct levels in Japanese Americans as compared to whites or Native Hawaiians. This data, combined with genotyping for polymorphisms in relevant enzymes, will provide insight into the ethnic differences in 1,3-butadiene metabolism, DNA adduct formation, and lung cancer risk. Understanding the mechanisms that determine cancer risk is vital to identifying high-risk patients for prevention efforts. Interestingly, we observed low but detectable amounts of Epiguanine 2 in the urine of non-smokers in this study. Our lab has previously discovered that individuals with no known exposure to 1,3-butadiene, unexposed laboratory animals, and untreated human cells in culture contain measurable amounts of Epiguanine 2 and another butadiene DNA adduct, THBG. This suggests the existence of endogenous and/or dietary sources of both adducts, which could contribute to DNA damage and cancer in unexposed individuals.
So a really long-term goal of this project would be able to take a sample from someone and tell them how likely they are to develop lung cancer. and be able to target the high-risk smokers for smoking cessation groups, and that's really a project that I want to be a part of.
In the third project, we are focusing on epigenetics. This is a relatively new area in my laboratory. We just published the first big paper in Nature Scientific Reports. And there we investigate DNA methylation and demethylation. DNA methylation occurs primarily at cytosine bases of DNA and is used to silence genes so that the patterns of gene expression can be established. So we know what proteins introduce methylation marks. Those are DNA methyltransferases. But for many years it wasn't known what proteins remove the epigenetic mark, this methylation mark from DNA, enable us to turn the genes back on. So it was found that 10-11 translocation dioxygenases play a very important role in oxidizing the methyl group within DNA to hydroxymethyl C, formyl C, and carboxyl C. And then these oxidized forms are recognized as DNA damage and are removed by DNA repair machinery. So this enables these genes to be activated by removing a repressive mark. So we are focusing on Tet proteins. There are actually 3 of them, Tet1, Tet2, and Tet3. And these are metalloproteins that use alpha-ketoglutarate cofactor to oxidize the methyl groups in DNA. So we want to understand the roles of these oxidized bases in DNA by conducting chemical proteomics experiments to identify specific readers of this hydroxymethylated, formylated, and carboxylated cytosines in DNA. And also we are looking in animal models to understand the effects of inflammation on epigenetic marks. So we have a large grant from the NIH to understand how epigenetics provides a link between inflammation and cancer.
My name is Jenna Fernandez, and I'm a graduate student in the lab of Dr. Natalia Tretyakova. So my research involves DNA epigenetic marks, such as methylcytosine and hydroxymethylcytosine. We're interested in the role that epigenetic deregulation plays in the early stages of lung cancer.
Lung cancer is a leading cause of cancer deaths worldwide, and over 80% of these cases are directly attributable to smoking. Cigarette smoke contains over 60 known carcinogens, as well as genotoxic co-carcinogens such as the inflammatory agent LPS. Epigenetic deregulation is one of the mechanisms contributing to the development of lung cancer. Epigenetic changes, such as aberrant patterns of DNA methylation and altered gene expression levels, have been observed in smoking-induced lung cancer. Work in our lab explores whether this epigenetic shift may be mediated by inflammation. Chronic inflammation is a major risk factor for the development of lung cancer in smokers. Furthermore, we have shown that inhalation exposure to inflammatory agent LPS induces early epigenetic changes in the lung, analogous to the changes observed in lung tumors, shown in these 2 graphs on the right. Thus, we hypothesize that inflammatory agents present in cigarette smoke cause chronic inflammation in the lung, and this inflammation induces early epigenetic changes that play a role in lung cancer development. To investigate the role inflammation plays in the development of lung cancer, mice are exposed to either LPS or cigarette smoke. At the end of our exposures, the mice are sacrificed. A subset of mice are used for pathology, and the blood is collected to measure nicotine metabolites and levels of inflammation in the blood. The remaining mice are used to collect alveolar type 2 lung cells. These cells have been shown to be precursors to pulmonary adenocarcinomas, which make up the majority of smoking-induced lung cancers. From these cells, we then isolate DNA, RNA, and protein. The proteins are used to study proteomic changes associated with treatment. The DNA is used to analyze the global changes in DNA methylation and hydroxymethylation, as well as the site-specific changes in the genome. The RNA is used to assess gene expression patterns. We first conducted RNA-seq to study the changes in gene expression caused by exposure to LPS or cigarette smoke. When we look at changes in gene expression associated with LPS treatment, we see an upregulation of epigenetic regulators, shown here. This is interesting because it may suggest that inflammation induces epigenetic change. This supports what I showed earlier, where the inflammation caused by LPS was driving the loss of hydroxymethylcytosine prior to tumor formation. Now, because we are interested in the role that inflammation plays in smoking-induced lung cancer, we want to determine if there are common patterns in gene expression between the LPS and cigarette smoke treatments. There are almost 1,500 differentially expressed genes in common in the two treatments. Looking at the functions of this subset of genes, I see a significant enrichment in genes associated with inflammation and carcinogenesis. These genes can serve as useful targets in our future studies to better understand how inflammation and epigenetics work together to contribute to lung cancer development.
This work is important because it gives us a better understanding of the mechanisms behind cancer initiation. And beyond that, epigenetics is very important and exciting because it shows us it gives us more information on how our environment and our lifestyle choices, how they can impact our health and how it affects which genes are expressed and our risk of developing disease.
In our fourth project, we investigate DNA protein cross-linking. So in the presence of biselectrophiles or free radicals, proteins can become irreversibly trapped on DNA. So we are interested in this process because formation of such DNA protein crosslinks appears to be associated with aging. So we know that as we age, specifically in the heart and in the brain, there is an increase in the number of DNA protein crosslinks. So my project, that is actually a collaboration with Colin Campbell in pharmacology, it aims to understand how these DNA protein crosslinks form and how they're handled by cells, and how maybe when this repair process fails, it could lead to aging and other diseases.
My name is Luke Erber. I'm a postdoctoral research associate in the Tet lab at the University of Minnesota. I am studying DNA protein crosslinks. These are bulky DNA lesions that inhibit DNA replication, transcription, and I'm studying how these bulky lesions are repaired. So DNA protein crosslinks are either formed at the epigenetic site or they block the activity of epigenetic readers and writers, inhibiting natural epigenetic processes. So it's important we understand how these DNA protein crosslinks are formed and what kind of exposures are leading to this increased risk of DNA protein formation. And we're also interested in knowing how these DNA protein crosslinks are repaired at these epigenetic sites so we can come up with new therapeutics in order to combat these lesions.
Very recently, with the COVID pandemic influencing the world, we also became engaged in investigation of potential inhibitors of COVID RNA polymerases. So our lab is known for our ability to synthesize unnatural nucleosides. And so we are making analogs of remdesivir, which is one of the therapeutic agents that's currently being investigated for treatment of COVID. And we are using the tools of molecular modeling, organic synthesis, and also biochemical assays to test the activity of these analogs in human cells. So what kind of links these different projects is our expertise in nucleic acid chemistry and also in mass spectrometry. My laboratory has two powerful mass spectrometers. One is a Quantiva triple quadrupole mass spectrometer, and the second one is a Q-exactive Orbitrap mass spectrometer that allows us for accurate mass determination and very high resolution analysis of biomolecules and small molecules. So each student who joins the lab has the opportunity to learn hands-on how to operate this instrumentation, and all of them apply mass spectrometry in their research.
Yeah, and to prospective students who want to join the Tretyakova group, I say if you don't think your interests necessarily line up with everything that we do, don't let that be a deterrent. You'd be surprised how much interplay there is between the different fields and interests. When I came in, I didn't necessarily know exactly what I wanted to do. I was pretty torn between, do I want to keep doing synthesis? Do I want to continue working on epigenetics like I had in the past, or do I want to try something entirely different? Given the lab is so interdisciplinary, there's so many opportunities to kind of explore and maybe try something new. If you would have asked undergrad me if I was going to become a bioanalytical mass spectrometrist, I would have laughed in my face. But here I am now running HPLC and mass spec almost every week. So don't be afraid to branch out and see where things take you.
So Dr. Trechakova is always open for questions or concerns. We have regular project update meetings, and so whenever things aren't going super well, because that happens with research, she always has a suggestion at the top of her mind for what to try next, which is very helpful in moving the projects forward and having them be successful.
My laboratory is probably a right fit for someone who wants to use the tools of chemistry and biology to be able to achieve a specific research goal. So we don't limit ourselves to just making a compound or just doing the biological studies. We are able to combine these two so the trainee can experience organic synthesis, solid phase synthesis, work with cell culture in animals, and also be engaged in human studies, all in one project. I think that's pretty unique, and so if that's something you are interested in, I hope you'll consider my group.